Hello, my name is Jaimi and I'm a postdoc in the Blackburn lab at the Florida Museum of Natural History. After a long COVID-induced delay, I started here at UF in January, and I've been working in cahoots with Dave Blackburn and Ed Stanley on the oVert project. So first, a little bit about the oVert project. It's funded by NSF and based at the Florida Museum of Natural History. And oVert stands for Open Vertebrate. And basically, the oVert team aims to facilitate the exploration of vertebrate diversity by CT scanning all of the vertebrates and making the data freely available for research, education, and wherever else they might be valuable. So in collaboration with over 20 other institutions, including 6 scanning institutions, we are generating CT data for both skeletons and soft tissue anatomy of specimens in natural history collections. These data sets are uploaded to the online repository Morphosource, where they are freely available for download. We also actively plan workshops, outreach, and create resources to help people make use of this data. So far for our skeletal scans, we have scanned over 50% of described genera, and we are closing in on finishing off the amphibians and reptiles. Obviously, our scanning efforts are still ongoing. The scans we have made available have been used in research with over 70 publications using data from Morphosource. And here are some examples of some of the educational activities and workshops that have been produced, either using oVert data or by members of the oVert team. So for the first stages that make up the oVert project, all of our data is in shades of grey, in the form of the stacks of grayscale images that make up a CT scan. It's not until the final stages that we make CT data explorable in an intuitive way by people who are not necessarily familiar with 3D imaging, so that they can be used for teaching and general exploration of vertebrate anatomy. So that's what I'd like to take you through today. So I'm going to take you through this workflow that I use with the end goal of making a scene using 3D models of vertebrate anatomy. So let's begin at stage 1, which is imaging hard tissues. To image hard tissues, we use a CT scanner. Here at UF, we have a Tommy scanner, which is located at the Nanoscale Research Facility, and this is where we do all of our CT scanning. So I'm going to take an animal that all of you in Gainesville have probably seen at some point, a green anole. And I'm going to go into the herpetology collection here at the Florida Museum and pick a specimen. We actually have hundreds of anoles here at Florida Museum, so I was able to choose a nice male that was fixed with his dewlap extended. So I'm going to take this specimen and put it in the CT scanner. And a CT scan is basically a series of X-ray images of a specimen that are all taken from different angles. And we can take these images and use computer processing to create cross-sectional slices of the specimen. And we end up with a data set like this. So with the help of X-rays, we have imaged the skeleton of our green anole and rendered it in 3D. So at the bottom, at the top, you can see a fly-through of the stack of grayscale images or tomograms that were generated from the X-ray images. And together, these images make up a 3-dimensional volume that can be rendered in 3D as shown at the bottom of the screen. And now that we have our skeleton data set, we can start to image the soft tissues of our green anole. To image the soft tissues, we use a method called diffusible iodine-based contrast-enhanced CT. So we're going to CT scan our green anole again, but this time we will stain the specimen with a solution of iodine. Now the iodine binds to carbohydrates and lipids in a specimen and increases the radiodensity of soft tissues in predictable ways. So here on top, you can see a rendering of our initial skeleton CT scan. You can see a lot of gray areas where there are soft tissues, but they all show up at a similar density, so we can't differentiate between different tissues or organs. Now at the bottom, you'll see a CT scan of the same specimen after it was stained with iodine for 6 days. Now we are seeing the same cross-sectional images, but this time you can see how the radio density of different types of soft tissue has increased in different ways. Now that we have both of our CT data sets, we can start doing a digital dissection. So we will perform a segmentation or a digital dissection on the parts of the animal that we want to build 3D models of. So here at UF, we use a program called VG Studio Max. And this is designed to work with CT volumes and define regions of interest and render them out of our stack of CT images. So we can scroll through our volume in different directions and identify the different structures that we might want to visualize. And I'm going to show you this using the DICT dataset because that's a little bit more exciting than the skeleton dataset. So here is a single CT slice or cross section, and in this slice I can see lots of different soft tissue structures. So we can see the brain with the olfactory bulb and the spinal cord. You can see the trachea, which leads into the lung, and there's even still a little bubble of air left in the lung. You can see the heart, you can see the liver, so the esophagus, which leads into the stomach and the gut. There is the pancreas, there's one of the kidneys, and there's one of the gonads. And we can even visualize the tongue. So if we manage to do what we've done here in all of the 848 slices that make up this particular CT scan, we end up with something that looks like this. We have defined our regions of interest and we are able to visualize them in 3 dimensions. So here I'm slicing through them in that same sagittal plane. And here I have made the rest of the body transparent so that you can see the organs in 3 dimensions. So right now we are still working with our CT data sets and we're still working in VG Studio. But what we've done here is used different rendering styles for different subvolumes of CT data. But in this form, not everyone has the resources or the expertise to use these renderings and volumes to explore anatomy. So I want to convert these structures into a form that can be used by everyone for many different things. And this brings me to the next part of the process. The fourth phase of this process is mesh generation. What we're going to do is take the structures that we're interested in and generate these objects called meshes. And the meshes basically represent the surface geometries of the structures that we want to visualize. So I am going to take my subvolumes here and I'm going to use a function in VG to convert each of these structures into a mesh. These meshes are basically each an individual 3D model, which represents the surface of that object. And now we can export these 3D models out of VG Studio, where they can be viewed and edited with other software and are much less computationally expensive to work with and view. So there are a lot of different programs out there for working with meshes, and many of them are actually free. My personal preference is this program called Blender. So here is my 3D viewer in Blender, where I have imported all of my animal organs as mesh files. And you can see the liver selected here. And when I zoom in on it, you'll see that the meshes are actually made up of lots of little triangles called faces. Now, instead of working with a 3D volume of CT data, we work with the faces of each mesh. And we can edit these faces to give them the look that we deem appropriate for the model that we are making. So up until this point, we have been working with shades of grey, but it is time for the fun stuff. We are moving into the mesh editing stage of the workflow. There are a bunch of different things we can do to our meshes to give them the look that we want. So for this part of the process, I mainly use Blender, although most of these things can be achieved in other mesh editing software. So after we export our mesh from VG Studio, it might look something like this. This is the gallbladder from our green anole. The mesh generating function in VG gives its best shot at making a structure from the region of interest that you defined. But I think we can make it look a little better than this. So we can smooth it out to make it look nicer and make the details of the structure more clear. We can edit the mesh. We can delete pieces, fill holes, move pieces, add more pieces. You can basically add or remove as many triangles as you want to perfect your mesh. Another thing we can do is simplify the mesh. This is especially useful for large models. Larger models can take up a lot of memory and they can be difficult to work with. They can also be difficult for others to make use of them afterwards. So you can decimate the number of triangles until you are happy with the size of your mesh. We can also start to add colors. So there are a couple of ways to approach this. You can paint the vertices, which are the points that are at the tip of each triangle on that mesh. And we can fill the entire mesh with color by assigning colors to these vertices. So we can assign a single color to a mesh, or we can use a paintbrush to paint those colors on to different areas. And another way to add color is to texturize your mesh. And this is where you take an image and you map that image onto the surface of your mesh. So in this case, I have taken an image of the texture of a head of broccoli, and I've mapped it onto the gallbladder here to show you how this might work. So here is an example of our green anole insides and their meshes, which have been cleaned up, smoothed, edited, and colored. In this case, I have used the Vertex painter and a combination of filling the meshes and painting different colors onto different regions. So now we're ready for the final step. If my mother was watching, which she is not, because it's 1 AM in Australia, I would tell her, please look away, because I'm about to make a scene. So now that we have made our meshes and edited them, we're going to put them together in an intuitive way. And this is the part where we make our models accessible for anyone that wants to see them. We use this website called Sketchfab, which allows the viewer to open the model and explore it in their web browser without any need for downloading any of the files or use any fancy software. All they need is a web browser and a link to the model. So now I want to show you a few examples of Sketchfab models that I have made for our Blackburn Lab Sketchfab page and some of the ways I add information to three-dimensional scenes. So this model is a leopard cat shark, which I've bisected or chopped in half to show a normal skull on one side and a transparent skull with the dentition shown on the other. So I've added a color key to give some information about tooth replacement in the shark. And I have also added a little shark silhouette and the name of the shark in the bottom corner to show the viewer what kind of animal they're looking at. We can also use Sketchfab to add annotations directly onto the model itself. So here is the heart and lungs of a Hermesis frog. And I've added annotations onto different parts of the anatomy here, which lets the user take a virtual tour through the anatomy of the heart and lungs of this frog and gives them some information about these structures. We can also add multiple models into the same scene and arrange them in a way that allows the viewer to view both and make comparisons between them. So in this case, not only will you see that I've added multiple models to the same scene, I've also added an object that I made in Blender. In this scene, you will see the three-dimensional evolutionary tree, along with the models of different vertebrate forelimbs. And the resulting scene is information rich, and it allows the viewer to take a tour into a system that is complex. Now let's come back to our anole model. I gave him some skin and I animated a virtual dissection of his body parts. So you can pause this animation at any time. So we can take our animal apart and we can pause the animation. It's about to pause, spin it around, look at the pieces, see what the different organs look like individually. And then when we're done, we can hit play and we can watch our green animal put himself back together again. We also have the option to pause this when the organs are put back together and the skin is still off to see how the organs are associated with each other. So one other very useful feature of Sketchfab is that we can create collections for models. So here's one example of a collection I've created, the Colors of Skull Anatomy collection, which is pretty popular in vertebrate anatomy classes. And I'm still working on more skulls for this. And basically, you can take a collection of models that are all used for a similar purpose or fit a similar theme. And then you can use the link to that collection to take a viewer to all of those models at once. So here's one last example of a model that has annotations. It has contextual information in the form of a color key and the picture of the tuatara at the bottom. And it has an animation. You can pause this animation at any time, so you can examine the individual bones that make up this tuatara skull, or you can examine the tuatara skull and how the pieces fit together. And with that, I will leave you with a taste of the models that I've created for the Blackburn Lab Sketchfab page, which I will drop a link to in the chat. Feel free to visit and explore vertebrate anatomy for yourself. And lastly, I would like to thank all of these people who have played some part in all of this work and the models that you see here. So if you have any questions, I will be happy to answer them.